Diploblastic and triploblastic organization. Animals in which cells are arranged in two embryonic layers and external ectoderm and internal endoderm are called as diploblastic animals. Example, coelenterids. So, see figure number 4.2 showing germinal layers diploblastic and triploblastic. So figure number A is diploblastic. So see the internal layer, that is endoderm. After that it is mesoglea, and third outermost layer, that is ectoderm. Endoderm, mesoglea and endoderm. So endoderm is the innermost layer, outermost layer is ectoderm. So the organisms in which two embryonic layers, that is external ectoderm, internal endoderm, are called as diploblastic animals. Diplo means two germinal layers, that is ectoderm, outer layer, endoderm, inner layer. It is called as diploblastic animals. And examples of these diploblastic animals are coelenterids. Coelenterids are the examples of diploblastic animals. Next is an undifferentiated layer, mesoglea, is present in between ectoderm and endoderm. So see in figure A, 4.2a, diploblastic animals. So we are seeing here outermost layer is ectoderm, innermost layer is mesoderm, and in between we are seeing in red color, that is mesoglea. This is called undifferentiated layer present in between ectoderm and endoderm, that is called as mesoglea. Next is the animals in which developing embryo has three germinal layers, mesoderm in between ectoderm and endoderm, are called as triploblastic animals. So see, we have seen outside layer is ectoderm, innermost layer is endoderm, in between ectoderm and endoderm there is a layer of mesoderm, and they exhibit three germinal layers, and they are called as triploblastic organism. The examples of these triploblastic animals is Platyhelminthes to chordates in figure 4.2b. Platyhelminth to chordates. These are the examples of triploblastic chordates. Next is coelom. Presence or absence of cavity between body wall and gut is very important in classification. So in body wall there is presence of cavity or not, it is very important for classification. The body cavity which is lined by mesoderm is called as coelom. See in figure 4.3a, coelomate. The body cavity which is lined by coelom, which is lined by mesoderm, is called as coelom. We have seen earlier about mesoderm, that is middle germinal layer, outer germinal layer is ectoderm, innermost layer is endoderm, and middle one layer is mesoderm. So the body cavity in which mesoderm is present, it is called as coelomate animals, means coelom. A body cavity in which it is lined by mesoderm is called as coelom. So see figure 4.3a, coelomate. So see the body cavity is lined by mesoderm, it is called as coelom. The next is the animals possessing coelom are called coelomates. Examples are annelid, mollusca, arthropoda, echinodermata, hemichordata and chordata. So from each step you have to learn examples in this animal kingdom. So we are learning the examples of coelomate. What is meant by coelomate? The animal which exhibit mesoderm in their germinal layers and they possess a coelom, and it is called as coelomates. The examples are Annelida, Mollusca, Arthropoda, echinodermata, hemichordata and chordata. In some animals, the body cavity is not lined by mesoderm. Instead of mesoderm, it is present as scattered pouches in between ectoderm and mesoderm. Such a body cavity is called as pseudocoelomate, and the animal possessing them are called as pseudocoelomates. Example, 𝐻𝐾𝐿. So what? In this pseudocoelom, figure number 4.3b, see in B diagram, what is shown there? The body cavity is not lined by mesoderm. Instead of mesoderm, mesoderm means what? Middle germinal layer. Instead of this middle germinal layer, there is scattered pouches in between ectoderm and endoderm. Such body cavity is called as pseudocoelom, and the animal possessing pseudocoelom is called as pseudocoelomates. So what we have seen in coelom, it has middle layer called as mesoderm and they called as coelom, and the animal possessing coelom is called as coelomates. In this pseudocoelom, there is scattered pouches instead of mesoderm, and such animal possessing pseudocoelom is called as pseudocoelomates. The examples are 𝐻𝐾𝐿. So the examples of coelomates was annelid, mollusca, arthropoda, echinoderm, hemichordata and chordata, and the examples of pseudocoelomates are 𝐻𝐾𝐿, only 𝐻𝐾𝐿. The animals in which body cavity is absent is called as acoelomates. The examples are platyhelminthes. So they may ask about what are coelomates. So you have to tell that the animal possessing coelom is called coelomates. And what is coelom? Coelom is a body cavity in which it is lined by mesoderm. Next is pseudocoelomate, the body cavity which is lined by scattered pouches instead of mesoderm. It is called as pseudocoelom, and examples of these pseudocoelomates are 𝐻𝐾𝐿 and acoelomate, the body cavity which do not present any of the coelom, means absent of body cavity is called as acoelomate. The examples are platyhelminthes. So the examples of coelomate is annelid, mollusca, arthropoda, echinodermata, hemichordata and chordata. You have to learn by heart all this. And pseudocoelomate examples are 𝐻𝐾𝐿 and acoelomates examples are platyhelminthes. They may ask about question from this diagram also. They may show figure number A and identify which diagram it is. So see the diagram of coelomates. So see the diagram of pseudocoelomate. They have shown the scattered pouches in middle layer, and see the diagram of acoelomate means absence of body cavity. It is called as acoelomate. So keep in mind they may ask question from anywhere of this NCERT. Next is segmentation. In some animals, the body is externally and internally divided into segments with serial repetition of at least some organs. For example, in earthworm, the body shows this pattern is called as metameric segmentation, and the phenomena is known as metamerism. So see, in some animals they have a body which is externally and internally divided by some segments with serial repetition of least some organs. For example, in earthworm there is a segmentation internally and externally, and this pattern shows metameric segmentation, and this phenomena is called as metamerism. So the metamerism is seen in earthworm. The example of metamerism is earthworm, or metameric segmentation is seen in earthworm because the body is divided internally and externally and serial repetition of least some organs. So they show the property of metamerism. The example is earthworm. Next is notochord. Notochord is mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. So we have seen about germinal layers. Diploblastic, triploblastic, the animals which possess external and internal germinal layer, externally ectoderm, internally endoderm, they are diploblastic. These are the germinal layers, and in between there is mesoderm, means in between ectoderm and endoderm there is mesodermal layer. So the notochord is mesodermally derived. So the notochord derived from that mesoderm. So it is called as mesodermally derived rod-like structure formed on the dorsal side of the embryonic development in some animals. Animals with notochord are called as chordates, and the animals which do not form this structure is called as non-chordates. Example are Porifera to echinoderms. So this notochord is present dorsally during the embryonic development. The animals which have notochord is called as chordates, and the animals which do not possess notochord is called as non-chordates. The examples of these non-chordates are Porifera to echinoderm. These are the non-chordates, means what? They do not have notochord, so they are called as non-chordates. Next is classification of animals. The broad classification of Animalia is based on some fundamental features are mentioned in preceding section is given in figure 4.4. Figure 4.4, broad classification of kingdom Animalia based on common fundamental features, different phyla are described. So from this animal kingdom, the most important table is given here. You have to learn by heart this table. In this table, if you are going to learn all these things, means it will be easier to understand and to remember all the examples. So they are given the kingdom. We have already known about this as we are studying about animal kingdom. So the kingdom is Animalia, and the first sentence of this chapter is kingdom Animalia. They are multicellular. So they are multicellular. Kingdom is Animalia and multicellular. And level of organization we have seen that cellular level of organization, tissue level of organization, and organ system level of organization. Earlier we have seen about the examples of this cellular level organization, that is sponges. Tissue level organization we have seen the examples like ctenophora, coelentrates, etc. Organ system level of organization we have seen earlier. So these are the level of organization. Next is symmetry. We have seen symmetry: asymmetrical, symmetrical, bilateral symmetrical, radial symmetrical. We have seen earlier. So they have shown asymmetrical animals, radial symmetrical, and bilateral symmetrical. Next is body cavity and coelom. We have studied earlier about body cavity. They are acoelomates. Then radial symmetry, there is acoelomate, and bilateral symmetry. They have shown about body without cavity, acoelomate, pseudocoelomate, and coelomates. In that acoelomate there is platyhelminthes, with pseudocoelomate aschelminthes, and with true coeloms annelid, arthropod, mollusca, echinodermata, hemichordata, and chordata. So you have to keep in mind this table is very important in that they have shown a level of organization, cellular, tissue, organ or organ system. According to that, we are going to study about cellular level organization. In that, the symmetry is means the animals which possess cellular level organization, they are mostly asymmetrical animals, means this symmetry is asymmetrical. We have seen the example of asymmetrical animal was sponges, because they divide because they do not divide body cavity into two equal halves, so they are called as asymmetrical animals, and their body cavity or coelom is acoelomata. Acoelomates, we have seen the examples of acoelomates. And next is phylum. Their phylum is Porifera. So the cellular level organization is seen in sponges, and this sponges is kept in phylum Porifera. Phylum name is Porifera. The next is tissue level organization or organ system level of organization is seen in symmetry, radial and bilateral symmetry. So in radial symmetry, the body or coelom is acoelomata. So the in radial symmetry, acoelomate animals, they are kept in phylum Coelenterata and Ctenophora. Coelenterata is also called as Cnidaria, the phylum Cnidaria. So acoelomates with radial symmetry, the examples are Cnidaria or Coelenterata or Ctenophora. Next is bilateral symmetry. In this bilateral symmetry, they have given uh, three types, in that acoelomates, pseudocoelomates and coelomates. So in bilateral symmetry, the acoelomate animals, acoelomate means what? They do not possess a body cavity. So the examples of acoelomate is platyhelminthes. With bilateral symmetry, the example of pseudocoelomate is ascalmanthus. Pseudocoelomate means what? With false coelom. Means false coelom means what? Instead of middle layer, that is mesoderm, there is a scattered pouches, so it is called as false coelom, pseudocoelomate. Example is ascalmanthus. The next is with bilateral symmetry, coelomates means true coelom. The examples are Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. So you have to keep in mind this sixth phylum, that is with true coelom. Annelida means 2A, then Mollusca, Echinodermata, then Hemichordata and Chordata. So you have to learn by heart this table. If you are going to learn this table, you are not going to face any difficulties in this animal kingdom. This is most important table for question point of view, exam point of view, and they may ask question from here. Examples of pseudocoelomate with bilateral symmetry, the answer is ascalmanthus. Or they may ask about examples of acoelomata with radial symmetry, the example is Ctenophora. Or the examples of acoelomata with asymmetrical symmetry, the example is Porifera. They may confuse in this, because in radial symmetry acoelomates are Ctenophora, Coelenterata and Cnidaria. In asymmetrical symmetry, the examples of acoelomata is Porifera. So they may confuse in this, because they are having different symmetry but acoelomates, and examples are also two different, so they may confuse here. So you have to learn by heart this table. Phylum Porifera. So we are going to learn about phylum Porifera. So the members of this phylum are commonly known as sponges. So from this phylum Porifera, we are going to learn about sponges, and they are commonly known as sponges. Means in this phylum we are going to learn about sponges. So see the figure 4.5, examples of Porifera. So all these are the types of sponges. Example first is Cycon, second is Euspongia, and third is Spongilla. So you may consider sponges also look like this. These are the examples of these sponges. So they are generally marine and mostly asymmetrical animals. So their habitat is marine, but they are asymmetrical. We have studied about asymmetrical symmetry. Means what? When the plane is passes between this body, between the when the plane is passes through the between the body, they do not divide into two equal halves, so it is called as asymmetrical animals. So the examples of asymmetrical animal is sponges. These are primitive multicellular animals and have cellular level of organization. So we have seen in level of organization that the cellular level of organization is exhibited by sponges and they are multicellular in primitive stage. So the next point about sponges is they are multicellular, primitively multicellular, and they possess cellular level of organization. So from starting onwards, the sponges are asymmetrical animals. They found mostly in marine habitat. Next is they were multicellular and they exhibit cellular level of organization. Next, sponges have water transport canal system. Water enters through the minute pores called as ostia in the body wall and central cavity called as spongocoel, from where it goes out through the osculum. So here the most important point is sponges are having water transport canal system. Sponges possess water transport canal system in that water enters through the minute pores called as ostia. So see the figure 4.5a cycon. So here they have shown a dots on the body of these sponges. So they are saying that the body wall is having minute pores. Through that minute pores, the water goes inside the body cavity. Water goes inside through the pores, and that pores are called as ostia. As soon as that water enters through the ostia inside the body cavity, and this body cavity is called as spongocoel, and the water enters the body cavity, and the body cavity is called as spongocoel, and the water comes out through that body cavity, it is called as osculum. Means when the water enters into the body cavity through ostia, and that body cavity is called as spongocoel, and when that water inside the body cavity comes out, it is called as osculum. So please read carefully about this. They have shown said that water enters to the minute pores. It is called as ostia. Means what? Water goes inside the body through ostia, means minute pores. They enter through the minute pores ostia in the body wall into the central cavity called as spongocoel. Means this water enters into the body cavity called as spongocoel, from where the water comes out, it is called as osculum. Means from the water enters the minute pores called as ostia, and this water in the body goes into the central cavity, and the central cavity is called as spongocoel, and from this spongocoel the water comes outside the body through osculum. So they possess a water transport canal system. Next, the minute pores ostia through which water enters into the body cavity, and this body cavity is called as spongocoel, and from this body cavity the water moves outside the body through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. So by this water transport system, this is helpful for food gathering, for respiratory exchange, and from removal of waste products. Next is choanocytes or collar cells line the spongocoel and the canals. Means what they are saying, choanocytes or collar cells is line the spongocoel. Spongocoel was body cavity, means inside the sponges, inside the body wall, inside the body cavity there is collar cells. body cavity is lined by collar cells or choanocytes. Choanocytes or collar cells line the spongocoel and the canal. Means inside the sponges, the body cavity is lined by collar cells or choanocytes. Next is digestion is intracellular. So the digestion in sponges is intracellular and the body is supported by skeleton made up of spicules and spongin fibers. So the body cavity is made up of skeleton and it is made up and body cavity is made up of skeleton with the help of spicules and spongin fibers. Spicules and spongin fiber is present inside the body and it is made up of skeleton. Next is sexes are not separate. Means what? In sponges sexes are not separate, they are called as hermaphrodite. Means what? Both the sexes are present in one organism, it is called as hermaphrodite. That is, eggs and sperms are produced by the same individual. Means what? Both the sex organs are present in same individual and the eggs and sperms are also produced in same body or same individual, so they are called as hermaphrodite. So keep in mind, they may ask about what is hermaphrodite, so you have to write there about the sexes are not separate, both the sex organs are present in same body and they release these gametes, means eggs and sperm, from the same individual, so they are called as hermaphrodites and their sexes are not separate. Next is sponges reproduce asexually by fragmentation. So the asexual reproduction in sponges is by fragmentation and sexually by the formation of gametes. Asexual by fragmentation, sexual by formation of gametes, then fertilization is internal and development is indirect, having larval stage which is morphologically distinct from the adult. Means what? Fertilization is internal fertilization is there and their development is indirect and they are having larval stage which is morphologically distinct from adults. Means what? Their larva means what? Their means larval stage means small stage of the sponges is distinct from adult. So the examples of these sponges are Cycon. Cycon is also called as Scypha. Spongilla, freshwater sponge. Euspongilla, bath sponge. You have to learn this example. You have to learn by heart this example. Spongilla, freshwater sponge. Euspongilla, bath sponge. Cycon, Scypha. So the examples of sponges are Cycon, Scypha, Cycon, Scypha, Spongilla, freshwater sponge, Spongilla's freshwater sponge, Euspongia, bath sponge, Euspongia, bath sponge. So let's revise these sponges. So keep in mind about sponges, they are marine, they are asymmetrical animals, they exhibit cellular level of body organization, they have water transport or canal system, and this canal system, the water enters through ostia and the central cavity is called as spongocoel and water comes out through the body through osculum. Next is they are having choanocyte or collar cells line the spongocoel. Next is digestion is intracellular and the body is supported by skeleton and the skeleton is made up of spicules and spongin fibers. In these sponges sexes are not separate, means they are called as hermaphrodite. Means what? Both the sex organs are present in one individual, that is eggs and sperms are produced by same individual. Next is sponges reproduce asexually by fragmentation. Next is sexually by gametes and fertilization internal, development is indirect and the larval stage which is morphologically distinct from adult. The examples are 𝐶𝐭𝐻, 𝐿-Spongilla and Spongilla. 𝐶𝐭𝐻 is also called as Scypha. 𝐿-Spongilla is also called as bath sponge, and Spongilla is also called as freshwater sponge. So figure 4.5, see the diagrams. A is 𝐶𝐭𝐻. So you have to keep in mind these diagrams very carefully, because they may give a diagram and they may ask you to label this or to give a proper name of this diagram or phylum of this diagram, or they may say about water transport or canal system is seen in which of this diagram. So you have to identify these diagrams very carefully. So to see the diagram number A, that is 𝐶𝐭𝐻, B is 𝐸-Spongilla, and C is Spongilla. So we have completed this phylum Porphyra. Next is phylum Coelenterata. Phylum Coelenterata is also called as Cnidaria. The next name of phylum Coelenterata is Cnidaria. They are aquatic, mostly marine, sessile or free-living, radially symmetrical animals. So they are marine, sessile, free-living, radially symmetrical animals. The name Cnidaria is derived from Cnidoblast or cnidocytes. Means what? The name derived from cnidoblast or cnidocytes, which contain stinging capsule or nematocytes. Cnidoblast or cnidocytes, they are given the name because they are having a stinging capsules and nematocytes present on their tentacles of the body. Means they are having stinging capsule present in their body, so it is called as cnidoblast or cnidocytes. The cnidoblasts are used for encourage, defense and capture of prey. So cnidoblasts are used for encourage, defense and capture of prey. Means they encourage the other organism, defense, and they capture the prey. Figure 4.7, they are given the diagram. So see diagrammatic view of cnidoblast. They are having one outer flagella, and see the body of cnidoblast. Cnidoblast is used for encouragement, defense and capture of prey. Then cnidarians exhibit tissue level of organization and are diploblastic. So we have seen earlier about tissue level of organization, and they exhibit diploblastic body form. So we are knowing about diploblastic. What is meant by diploblastic? The organism which have two germinal layers, and one is outer ectoderm and inner mesoderm. So these animals are called as diploblastic animals. They have central gastrovascular cavity with single opening mouth on hypostome. So they are having single gastrovascular cavity and with a single opening mouth on hypostome, means present above the body hypostome, and they are having single gastrovascular cavity with single opening. Then digestion is extracellular and intracellular. So here in cnidarians or coelentrates, the digestion is extracellular or intracellular. Both type of digestion is seen, extra and intra, in coelentrates. The most important question from here is they are having extra and intracellular extra and intracellular digestion. Next is some of the cnidarians. Example, corals have skeleton composed of calcium carbonate. So in some of the cnidarians, corals, the skeleton is made up of calcium carbonate. They may ask question about this. Previously asked your question. They are having corals. Some of the example of this cylindrite is corals, because the skeleton is composed of calcium carbonate. Next is cnidarians exhibit two body forms called as polyps and medusa. They are having two body forms, polyp and medusa. Figure number 4.6, cylindrita indicating outline of their body form. First is Aurelia medusa and next is Adamsia polyps. So Aurelia is also called as medusa and Adamsia is also called as polyps. So see figure A, it is Aurelia, and figure B is Adamsia. In this Aurelia, see the body form. They are showing umbrella like body form, and B is Adamsia, and it is also called as polyps. So let's see the cnidarians exhibit two basic body forms, polyps and medusa. The former is sessile and cylindrical like hydra and adamsia, whereas latter is umbrella shaped free swimming like aurelia and jellyfish. So see about adamsia. The adamsia is like sessile or cylindrical body form. So see in diagram B, they are having a cylindrical body form and it is sessile or cylindrical body form, and they show like their body form is like hydra and adamsia. So they show cylindrical and sessile body form like hydra and adamsia. And next is umbrella shaped and free swimming like aurelia or jellyfish. So see aurelia, figure number 4.6a, it is like aurelia and jellyfish and it is free swimming. Show free swimming like aurelia and jellyfish, it is umbrella shaped. Then next is those cnidarians which exhibit both forms exhibit alternation of generation, metagenesis, that is polyps reproduce medusa asexually and medusa produce polyps sexually. Example obelia. So keep in mind that they show alternation of generation, and this alternation of generation is called as metagenesis. That is polyps produce medusa asexually and medusa produce polyps sexually. The example is given obelia. The examples of this cylindrita are Phacelia, Portuguese man of war; Adamsia, sea anemone; Pinnatulla, sea pain; Gorgonia, sea fan; Mendrina, brain coral. So here there is no trick to learn this example. You have to learn by heart each and every example from each phylum, because they are mostly focusing with these examples and they are going to confuse you. So keep in mind you have to study and learn by heart all these examples. Examples are Phacelia. Phacelia is also called as Portuguese man of war. Next is Adamsia, sea anemone; Adamsia, sea anemone; Pinnatulla, sea pain; Pinnatulla, sea pain; Gorgonia, sea fan; Gorgonia, sea fan; Mendrina, brain coral; Mendrina, brain coral. So let's see from beginning onwards about cylindrita. They are aquatic, mostly marine, sessile or free swimming, radially symmetrical animals. So radial symmetry is seen in cylindrita. The next is the cnidaria, name derived from cnidoblast or cnidocytes, which contain stringing capsules or nematocytes present on the tentacles of the body. These cnidoblasts are used for encourage defense and capture of prey. They exhibit tissue level of organization and they are diploblastic organism. They have central gastrovascular cavity with single opening, mouth on hypostome, and their digestion is intracellular and extracellular. Some of the cnidarians, example corals, their skeleton is composed of calcium carbonate. Then next is they have two body forms, polyps and medusa, and they the former is sessile cylindrical like hydra and atomcia, whereas umbrella shaped and free swimming like aurelia and jellyfish. And they show alternation of generation metagenesis, that is polyps produce medusa asexually and medusa produce polyps sexually. The examples are Phycelia Portuguese man of war, Adamsia sea anemone, Pinnatula sea pain, Gorgonia sea fan, Mandrina brain coral.